All right. So today we're looking at day three of mitosis. These slides will be a little bit long, but there'll be several slides that we'll be going through. But that's because we do have a lot of images inside of this one, but it shouldn't be that bad. So mitosis, specifically, what we're looking at the stages of mitosis. Remember, mitosis is used for growth and repair. That's what we talked about last class. Produces two new cells that are identical to the parent, that's the original cell. Remember, that's what this unit is all about. We're producing identical daughter cells, identical offspring, whatever it is, it's identical to it. The cells are diploid, which means that we would say that their chromosomal number is 2n, to be that complete copy. So this right here is basically what some of the things we're going to be looking at. This is chromosomes during and what it would look like during metaphase of mitosis. Now, in this first image that just came up here on the bottom, if you notice these green things that are starting to show on the sides a little bit, well, I know that that's part of the spindle fibers. But I also see that small circle on the inside of it, and I know that small circle on the inside of the cell is going to be the nucleus. But I can't quite make out the chromosomes completely. So that tells me that it's really hazy. And because of that information, because I know that the nucleus is still intact, and that DNA, that chromatin, hasn't actually condensed to make chromosomes yet, I'm able to know what phase I'm in. So I'm not right here. I'm actually starting to get in that early prophase side of things because it's getting ready to start its process, but it hasn't really kicked off yet. Well, once prophase is over with and it's done everything it's supposed to do there, I know that then eventually the chromosomes are going to line up in the middle of the cell. If you notice these green lines, it kind of looks like a basketball. These green lines are attaching at the middle of these chromosomes. And so I know that whenever that happens, that only happens during metaphase. And now I start looking at these, and now these chromosomes, the chromatids are actually separated. The chromosomes are no longer in the middle, they've been separated. They're going to either side, they've been pulled apart. So that tells me it's anaphase. And then right here, they're all the way on the other side. The spindle fibers are almost gone, and we're starting to get this indentation. Well, now I know where I'm at. I'm at telophase. And lastly, now I have two completely different cells, and they both have a nucleus of their own. Well, that means that now we have completely split the cell apart into cytokinesis, and now we have two daughter cells. Now, what I just did for you there, and I know all that information wasn't up there, but what I just did for you is I just looked at these images, I described what was happening, and based on that, I could tell you what phase they were in. And that's exactly what you're going to have to do on this quiz and with this information. You're going to have so many questions that deal with your ability to know the phases and know what's happening. All right? So you need to really make sure that you study not only the images, but the information as well. So, like we said before, mitosis is the division of the nucleus. It is only going to occur in eukaryotes because we're talking about those somatic cells like the skin and bone. It does not occur in all cells. It does not occur in your brain cells, your muscle, or your sex cells. Sex cells do divide, but they actually do it through meiosis, and we'll be talking about that in our next unit. And of course, here's some pictures for you. Now, if you could take a second and try to think about which ones they might be, I'm going to go ahead and click over to the next one. So our four phases are prophase, metaphase, anaphase, telophase. So see if you know what that top left-hand picture is. Whenever I look at that, what I see is I see a bunch of gummy worm looking things all crowded together in the middle. Well, whenever they're all crowded together in a big ball like that in the middle, I know that division hasn't started happening yet, so that has to be prophase, and because prophase is first. Now, if I go down to the one right below it, that kind of looks like somebody went to the doctor and the doctor did a really bad job of stitching them up. because you have all those individual chromosomes that are all crammed up there right in the middle, lined up in a row. Well, because I know that that's what metaphase is, I know that's one metaphase. And then if I go to the top right one, I see that now has been separated, and now I have chromosomes on either side. And that's those individual chromatids. Well, I know that being pulled apart starts with an A part, and that is anaphase. And lastly, telophase is at the bottom. This is where I start to see the chromosomes separated into two different areas, forming two nuclei and starting to form two cells. So that tells me it's telophase. So let's go through each one of these, and so that you know what information you have to know about it. So prophase. Prophase is whenever the nuclear membrane and the nucleolus is broken down. They disappear. You no longer see them. The chromatin condenses and is clearly visible so that you can actually see chromosomes. Some pictures may be like artist renderings of them to where it look like, the X's. And then some of them might be actual pictures of it to where it just looks like gummy worms. But that's okay. The spindle fibers they're shown at the beginning, and then they start to connect at the centromere. That's the center. That's where that X comes together there in the middle. And it connects there on both sides. This is the longest phase of mitosis. Please do not get that confused with interphase. Interphase is the longest phase of the cell cycle. Prophase is the longest phase of mitosis, though. So this is an image kind of showing you where it's at. So, of course, we have our DNA that's on either side that's in both chromatids. And then that centromere is there connected at, the spindle fiber is connected at the centromere on the chromosome. Don't forget that spindle fibers are made of microtubules. We actually went over that in our unit on cells. So right here I have an image of prophase for you. So whenever I look at this, some definite signifiers for me is if you look in the middle where you see the chromosomes, the different X's in different colors, you see how that it looks like it's a broken dotted line that circles it? Well, that's telling me that the nucleus is breaking apart. Also, we see these centrioles, and they actually have it moved to the either side, and that is the centrioles. That's those little, cylinder shapes. They're on either side that kind of looks like they're the clouds for the rainbow happening. And then, of course, those lines going across, those are the spindle fibers. Now, they haven't quite yet connected to it, but they're about to connect to it. By the end of prophase, they'll be connected to the centromeres of the chromosome. Metaphase. Metaphase is really short. Chromosomes are moved to the center of the cell. The center of the cell is called the equator. So if you look at this, it's moved to you could also think of it as the middle of the cell. They're lined up in the middle. They're lined up in the center. We also refer to that as the metaphase plate. Now remember, what's going to happen at both places where you see these centrioles? These are going to become two cells. Remember that those chromosomes, those are actually two identical strands of DNA that are inside of those chromosomes. And so the reason why they're getting pulled to the middle is for this next part to happen. Sorry, there's just another picture of it. This is an actual picture of one under a microscope. And you see how they're lined up in the middle. Is anaphase. So now, in each one of these chromosomes that are being pulled apart, that means that now each side of this cell is going to have an identical copy of that DNA. Remember that whenever they're separated like this, or the individual sticks of the chromosome are called sister chromatids. So whenever the sister chromatids are separated and pulled to opposite poles, that's to allow the new daughter cells that's going to pinch off in between there. That's to allow both of them to have the same amount of DNA and it be identical to each other. If there's a mistake that happens in this one, that's where you could get some major issues, mainly whenever we're talking about in meiosis with non-disjunction there. Here's another picture of anaphase happening inside of a plant cell. The reason why I know it's a plant cell is you look at this, it kind of looks like a brick wall and how it's all shaped. And that tells me right there, because of the shape of it, it's definitely not an animal cell, and that is a plant cell because that's a cell wall that's making those shapes. And then we have telophase. Telophase is the exact opposite of prophase. Instead of the spindle fibers forming, now they're going to be disappearing. The nuclear envelope starts to form around the sister chromatid, so now we're starting to make a new nucleus. The nucleolus is going to reappear inside of that nuclear envelope, and the chromosomes are going to uncoil and reappear as chromatin, so they're no longer going to be tightly coiled up together. And so right here is where we start to see that. So remember, anaphase, these individual chromatids were separated, and now we're starting to build our new nucleus around these chromosomes. And if you look at it, they have an identical chromosome on both sides. And eventually, these centrioles are going to disappear, and a new nucleolus is going to reappear. Now, if you notice, you see how this is starting to cave in on the top and cave in on the bottom? That's because it's about to pinch apart. The process of the cell actually separating into two daughter cells is called cytokinesis. That's where that cytoplasm finally divides and breaks apart. Now, a lot of things teach this as, you can find some stuff that'll teach us as a completely different stage, and some just talk about this as happening at the end of telophase. They're both right. This does happen at the end of telophase. It's a very small part of it, but it's such a critical stage for it to separate. Plant cells, how they do theirs, because they don't pinch in because they have the wall, they actually form what we call as a cell plate. And that cell plate will eventually become thicker and harder and become the cell wall. In animal cells, whenever we say it pinches in, we're saying it's forming a cleavage or a furrow. And I have an actual image to show you what that will look like. This right here would be the cleavage or the furrow of an animal cell. And this would be the cell plate of a plant cell forming. Okay? Please don't forget that whenever you go through this process, you're creating a diploid cell. That the offspring are the exact same as that original parent cell. That those daughter cells are the exact same. Same number of chromosomes, and the DNA strands themselves are the exact same as each other. So everything's identical in this process. So what are the four phases? Prophase, metaphase, anaphase, telophase. Make sure you can describe the characteristics of each one and be able to illustrate it. We will be doing this in class a good bit. You'll have some activities that you have to do at home. How many daughter cells are produced? 2 daughter cells are produced. What's the final stage of the cell cycle? See right there where it says cell cycle? I know they're talking about cytokinesis. How does an animal cell divide versus a plant cell? Well, an animal cell is going to form that cleavage or furrow, whereas a plant cell is going to make that plate. What structure connects chromosomes to pull them apart during anaphase? We're talking about the spindle fibers is the structure that is connected to the centromere so that it can be pulled apart, so that chromosome can be pulled apart during anaphase. All right, so next class, what we're going to do is we're going to be reviewing this information. Now, don't forget, this information is so important to know because in our next unit on meiosis, part of our standards for meiosis is to be able to cite the differences between mitosis and meiosis. So, everything you have learned today and about mitosis and in this unit, you are going to desperately need in your next one as well, or you will do very poorly on answering those questions.